Okay, let's see how this works. So hopefully you're all turned in and ready to learn. Here are some basic rules as you're going through this. So hopefully this is going to work out really well. It gives me a chance to actually give you a little homework so you're watching the presentation so that you just don't take notes off somebody else or just skip right through it. So hopefully you'll have some fun with it and this will work pretty well. This is my first time recording something, so let me get my head straight and make sure everything's intact. So here are the rules. Anytime you see this little guy, wherever you want, there he is, you're going to want to take out your pencil, take out your paper, and you're going to write down the question that's on the slide or the question I tell you out loud that you're going to want to use. Once you get that question written down, then you're going to look up the answer. So I may actually tell you the answer on the slide, or I may have told you the answer on the slide before, or I might have you go to Google or some kind of search engine and look for the answer. And then when we meet next Monday, you're going to turn that paper in with all of the questions that have this picture specifically. So this guy. All right. There are some other questions. There's kind of review questions from last week to make sure that you're still on track, and I'll point those out too. But the ones that you want to write down are the ones that actually have the guy with the pencil and say write it down. All right. So let's get started. I think we covered all of them of allergy. I know that, and hypersensitivities. We covered autoimmunity. I think we covered blood types. We talked about transfusion issues and graft versus host transfusion. And now we're on to infection. So infection is kind of fun. What's interesting about infections is that most of the organisms that can infect you are already on your body, or you have exposure to them. When you look at your intestinal flora, or the flora is talking about the bacteria that's in your gut, you actually have about 10 trillion bacteria in your GI tract. If you remember, like the first week of class or from physiology or anatomy, I know in physiology I always talked about it, and your anatomy instructor probably talked about it too, but you have about 1 trillion cells in your body. Those 1 trillion cells, you have 10 trillion more bacteria. That means the bacteria in your body actually outnumber you 10 to 1. There are about 30,000 different types of bacteria that are living in your GI tract. And typically, they're what we call commensal organisms. They live with us, they live in harmony, they're comfortable with us, and we work together. And we'll talk about these 4 different types. Actually, I'll have you jot down the 4 different types that are in your textbook. But most of these organisms are there actually to help us while we help them. So it's kind of a mutualistic relationship. Like I said, like E. coli. E. coli gets a bad rap. It seems like every year, for the last 3 years, we've had these big outbreaks of E. coli on spinach, and it makes people sick, it makes some people die. Typically, the people you hear getting sick and dying are the young, like small children with weak immune systems, the elderly that have weak immune systems, pregnant women that have compromised immune systems, or people that are just at risk in general. It's typical as people that have a weak immune system because E. coli is a natural part of our intestinal flora. It helps us to break down nutrients and release vitamin K that's important for blood clotting. We need E. coli in our system to help us with those things. In fact, what they've done is they've done research where they take a mouse, and when the mouse is born, they have it in a sterile room. They sterilize the mother, they sterilize the baby, and these mice have no exposure to external germs. They're called gnotobiotic mice. They're totally germ-free, and they find these gnotobiotic mice, they're actually not all that healthy because they need those natural bacteria inside their intestine to release nutrients. So it's kind of interesting if you ever get a chance to Google it. It's spelled Gnotobiotic, mice. Gnotobiotic mice. They're germ-free. They actually raise them, and they'll introduce 1 bacteria at a time to see what that bacteria does for us. And they do this for a couple reasons, because there's some bacteria that live in your intestines we can't actually extract and grow them on a petri dish because they're ideally grown in your intestines. You bring them out of the intestines and they die. They can't live. All right? So some of those normal flora, they help us. Sometimes these organisms that were working with us, though, if we get sick or we give them an opportunity, like a break in our GI tract, like an ulcer, or some kind of penetration, like an appendix burst, they get into our tissues and they become what we call an opportunistic microorganism. So opportunistics, they become pathogenic. Pathos, of course, means suffering or disease. Genic means generated. So what they're going to do is generate suffering, disease, or diseases in general. So some of those things like E. coli, when they become opportunistic, they can actually make us really sick. So here's just an example of your gut bacteria. You can see here are your, excuse me, here are the cilia, excuse me, inside of the intestinal tract, and here's all kinds of different bacteria. They've actually labeled these bacteria so they could identify different types, and these are just a few types of that bacteria, the ones that have specific genes inside of them. You can see them all along here. And here you have the villi, the cilia, the folds. And along there you have the mucous membrane. So you can see the difference. And there's so much GI bacteria inside of you that when you go to the bathroom, number 2, and you turn and look, about 60% of what's in the toilet is actually bacteria. So the bulk of your feces is actually bacteria. For some reason this isn't clicking. Had a better, there we go, better picture. So this is a mouse intestine. You can see some of the villi and the folds. Here you can see some bacteria and some of the food substances in it. So this mouse gave its life so that we could take a look at its intestines. So next thing, what you want to write down, and here's the first write it down. You're going to write these 4 things down. So first the question, what are the 4 relationships between human and bacteria, and what do they mean? And I don't remember what page it is in your textbook, but you want to look at relationships like what do we call it if only the human benefits? Or what do we call it if only the bacteria benefits? Or what do we call those disease-generating organisms? So you want to write those 4 different types down and what they do, or what makes them unique. There's actually a box in your textbook that you'll look for that has all 4 of them identified. So I'm not trying to make it tricky or hard. I just want you to know the 4 terms. All right. So pathogens. What's interesting is this bacteria in your gut, it's good for you. What's also interesting to look at is that these things that generate disease in us, they have creatures that generate disease in them. And then those creatures have pathogens, and those creatures have pathogens. So if you look at a structure like a flea as an example, there's a flea sitting on top of somebody's fingernail. And when you look at that flea and you zoom in on it, where'd my slide go? There we go. You can see the structure of the flea. Look at those legs. It's incredible the way that they can bounce. But if you zoom in on just the leg area, you can see that the leg of the flea actually has a mite. It's a parasite that's living in the flea. So if this mite kills, if we die, then this mite's going to feed on the flea and kill the flea. But then what about the mite? If you look even closer, the mite actually has bacteria sitting on the surface of it. And if you look a little bit closer at that bacteria on the surface of the mite, you can see some here, a little bit there, some there. You can see all these little bacteria sitting on the face of the mite. And yeah, that's what that mite looks like up close in the face. And then when you look at the bacteria, the surface of the bacteria actually have pathogens. So one of our pathogens, things that cause disease in us, has its own pathogen. That pathogen has a pathogen. And even that bacteria, the little teeny tiny bacteria you see in this picture, they have their own pathogens, viruses, called bacteriophages. They get in, they destroy the bacteria, they kill the bacteria and cause disease in the bacteria. When you look at those bacteriophages, they can actually have a pathogen. So a prion, and with prions we'll talk a little bit about mad cow disease, or we did, actually we already talked a little bit about mad cow disease. But these prions, they can affect the proteins in the virus. Remember, the virus is a long structure of proteins around its outside membrane. These prions can destroy it. And then prions can be attacked too. When we talk about free radicals are just little molecules that are unstable. And when they attack another molecule, they cause a chain reaction. So this free radical can actually attack that prion. And then you can basically go into infinity, smaller and smaller structures. And now that we've discovered the Higgs boson, who knows how far down we can look at disorders that are caused by basically physics rules. So you want to know the stages of infection. When you get an infection, there's actually this initial phase which is called the incubation stage. So as you're incubating, you're growing this stuff, it's kind of silent. They get into you and they colonize. Like, even for instance, when you eat like yogurt, there's a lot of debate on how much of the yogurt's bacteria gets down into your GI tract, but the consensus is that you get a lot of yogurt with a lot of bacteria, and some of that's going to live and make it all the way down into your lower intestines. At that point, they colonize, they start growing, they start increasing. These are mutualistic. You're feeding them 3 times a day. You're giving them a warm, humid environment. It's perfect for growing the bacteria. And in return, they'll break down nutrients. Like, for instance, people with lactose intolerant, they have a problem breaking down lactose. But what's interesting is they can eat yogurt, and they can tolerate yogurt better because the bacteria in yogurt actually break apart the lactose for the person. So that was another example of those mutualistic relationships. All right, but colonization, they get inside of us. They start setting up a home. Here's where it becomes kind of problematic. They can invade. If you open up this opportunity for them to get in, they invade into the tissue, they'll start multiplying, they'll start rapidly reproducing, and then they'll start spreading to different areas. So clinically, we refer to some of these stages as, like I said, the incubation stage. It's kind of a silent stage. Okay? The pathogen, you got entry into the host and it starts replicating. You have the prodromal phase, which is before basically the big hit, before they start getting all these clinical symptoms. They may get a little itchiness, a little runny nose, maybe some dry eyes. And then they get this like peak clinical stage where they get aches and chills and vomiting. And then ideally, when you get done with it, you have this recovery stage where you bounce back from it. So these are the stages of infections, and I kind of told you the clinical phases that go right along with them. So the next thing we want to be able to do is describe the factors influencing infection by a pathogen. One of them is the mechanism of action. Mechanism of action is how they actually damage your cells. Does this invader actually attack your cell and get in it and destroy the cell? Or does the invader dump some kind of toxin in the environment that kind of attacks the cell so that the invader can run away, basically? It's kind of like a smoke screen for the invader. The invader throws a smoke screen out that damages your cells, so the invader doesn't actually have to make contact with your cells. Okay. Next term you want to be familiar with is infectivity, and that's the ability to spread, invade, and multiply in this specific host, so in you. If it has high infectivity, it has a high ability to invade and multiply in you. The next term is pathogenicity, and that's how fast they can make damage or produce toxins. So some bacteria can make toxins really, really quick, faster than your immune system can clean it up, and that's when the problem really hits. Others are really slow at making toxins, and your body can fight that infection, take care of the toxins, and kill the bacteria before you start seeing real clinical stages. The next term is virulence, and that's the ability to cause harm. So, in other words, you might want to think of it as the number of organisms it takes to kill the host. Like, we're talking about E. coli. E. coli's got to take thousands or hundreds of thousands of E. coli to actually kill you or attack you. And when you talk about something like the Ebola virus, then it only takes a couple of those pathogens to get into you to actually kill you. So, pathogenicity, virulence is how able it is to kill you, or the number of organisms it takes to kill you. Immunogenicity, this is how fast they start your immune response. Some bacteria or viruses are good at hiding. They don't trigger your immune response right away. They can hide very deceptively. So when you think of something like HIV takes a long time to actually be detected in your system because it's pretty good at hiding, pretty good at camouflaging itself in your system. So in some cases it can take 4 to 7 weeks before it actually shows as HIV positive, where your body is showing a large immune response. Other times it can take over a year. So it has kind of a low immunogenicity. Next one is toxogenicity, so how easily it makes toxins. And we'll talk about endotoxins and exotoxins. Basically, how easily it makes these things that can harm you. So toxogenicity directly affects its virulence, how easily it causes harm. And then port of entries, how they get in. So is it through contact? Is it through inhalation? Is it through ingestion? Is it because of a bite? So when you think of something like rabies, a dog bites you, or a raccoon bites you, or a bat bites you, or whatever, that's its route of entry. That's how it got into you. The most common routes of entry are actually through the respiratory system, the GI tract, or the urogenital system. And primarily the respiratory and the GI tract. And when you think of that, you want to think of the GI tract. When you get in the small intestine, it's really a delicate layer. It's very thin. It's not as toxic as the stomach. It's a very mild environment. It's slightly alkaline, so it's not super toxic. And the pH isn't that dramatic. But the layers, it's designed for absorption. So if a bacteria gets in there, it could potentially get in. The same thing with your respiratory tract. When you breathe in, the alveoli are super thin. A bacteria has a higher risk, or a virus has a higher risk of getting in through that route. Same thing with when you're breathing in and the virus or the bacteria gets into the top of your nose. All the way in the top of your nose and the back by the cribriform plate, there's a pathway that can actually get right up into the brain. So when we get into the nervous system next week, we'll talk about things like encephalitis or meningitis and how they're affected by things you can be inhaling all the time. And did I miss any? Communicability. So communicability is how easily it's spread from one person to the next. Right? When we talk about infectivity, it's how easily does it get into you. Communicability is how easily does it get from you to your neighbor to the person after you. And when you look at communicability, it's actually different depending on the regions of the world. When you're looking at something like a third world country or an underdeveloped country, the main route that these things travel is through water. So soiled water, dirty water, polluted water. If you're looking at streams, one person's defecating upstream and another person's drinking it downstream, or they're bathing in it downstream. When you're looking at a highly developed country like the United States, the primary way that things get moved around is by touch or by sneezes. So when you aerosolize these things. So when you think of a sneeze, how would that affect the portal of entry? How is it getting into the next person? They're breathing it in, right? So it's the respiratory tract. Okay, so this question, and you don't have to write this one down because the little guy with the pencil's not on this, but when you think about this, tuberculosis, and we're going to talk about TB just a little bit more in a couple more slides, but it's caused by a germ called the Mycobacterium tuberculosis, and then tuberculous bacterium, which is TB. Its main activities produce these biochemicals. It produces these little structures that are released into the environment, and they kind of distract your immune system. So this TB is really sneaky. What it does is it sneaks into your white blood cells, it hides inside of them, but before it did that, it dumped a bunch of this chemical in the environment to distract the other white blood cells so it gets a chance to sneak in the back door of a white blood cell and hide inside of it. So I'm trying to avoid a word, and it's kind of hard to tiptoe around it, but so when we refer to this factor for infection, what would it be? When they release something into the environment that affects the host. It's called toxogenicity. How easily it makes this toxin or this chemical that affects the other cells. So here are a couple classes, and you don't have to go into great detail right now because I'm actually going to give you a little bit of homework assignment on these, but here's some general classes of infectious microorganisms that your textbook goes into. So when you're looking at some of these things like a virus is not living, and that's something you want to think about. A virus can't replicate on its own. How viruses affect you is they'll get into your cell, they inject either RNA or DNA, which we've talked about before, into the cell, and they alter the genetics. Right? So you already know pretty much how a virus works. And again, you want to remember, we really pushed this last week, is that once a virus is in you, can you get rid of it? Now, it's hard to get rid of it because it's in your cells. The only way to get rid of it is to kill off all the cells that have the virus before the virus can spread to other areas. Next one's some things like chlamydia, rickettsia, mycoplasma, bacteria, and on the next slide I'm going to have you look up some of these things. Excuse me. But when you look at something like a mycoplasma, it has no cell wall. Pneumonia is a really good example. There are several causes of pneumonia, but one of the causes of pneumonia is a bacteria with no cell wall on it. So it can kind of sneak around all over the place. Luckily, in some cases, it's easy to attack. Right? A mycobacteria has a real thick cell wall. In fact, it has a really thick acidic cell wall, and tuberculosis is one of those. So tuberculosis has a lot of weapons to sneak through your body. So it's super, super sneaky. Another one's fungi. And when you think of fungi is a eukaryote. When we're talking about viruses, it's not even a living cell. When we're talking about bacteria, it's a prokaryote. So fungi are eukaryotes. When you're attacking a fungi or trying to kill these fungi, you want to remember a lot of the properties of the fungi are very similar to human cells. So in the back of your mind, you have to think, how easy is it to kill these things? And if you're taking a chemical that's going to kill another eukaryote, what are the possible effects it could have on you? When you look at protozoa are technically one-celled animals, like Giardia, that lives in a vagina. So Giardia is a one-celled organism. It's actually a little tiny parasite that moves around or can swim around in the vagina. Right? And then this last group are kind of interesting. The helminths. helminths are basically parasitic worm-like structures. So when you're looking at a couple of these pictures, this one on the top up here, this is actually Candida albicans. This is a fungus. Right? When you're looking at the bottom one, this is trichinosis. This is a worm or parasite that you get from eating undercooked pork. And what it does is, this is a picture of skeletal muscle. You can see all the little worms inside of there. They've gotten into you, and then they get into your skeletal muscle where it's nice and warm and comfortable, and there's plenty of sugar and plenty of oxygen, and they're happy, and then they make a home there. Right? In fact, this is kind of interesting. On the next slide, like I said, I'm going to have you go through and do a little homework on this, but when you're looking at these things, here's an example of a helminth. In a human, it's a tapeworm. So this tapeworm is kind of interesting because if you look at it, it has this sucker structure on the front of it, and they call it the scolex. And this sucker structure attaches to your intestinal wall. Right? So what happens is you actually eat this egg. When the egg gets in you, your digestive tract opens up the egg, pops it open so that you can process it. And then this thing starts growing. It attaches to your wall. It starts feeding, so there you can see where the head attached. Basically it has these suckers and the hooks. And it starts eating on you. And it gets longer and longer and longer. So it just keeps getting longer. These little, structures, the proglottids, they're little sections. And the longer this worm gets, the very end ones start loosening up. So this is a really strong structure, like a string, or basically a living rope almost, that's held really tight. But at the very end, these will start breaking free, and these are full of the eggs. So it helps them reproduce. You have this thing growing in you. It can be several feet long. If I remember right, they can be up to about 18 feet long. And they just twist and wind through your intestines. And then when they're ready to release, they release these little egg sacs. They go off in your feces that drops on the ground, evolutionarily wise, we're going on the ground. And then it grabs a hold of its next victim. And it just moves from one body to the next to the next. One of the interesting things about this is that they release a chemical so that you'll usually only get one at a time. They're very territorial. And let's see, another interesting thing, this is a fun fact, and this isn't going to be on a test, but somebody determined a long time ago that, well, these things, they don't feed on you. They feed on the food that you're absorbing. So they eat your food instead of you absorbing the food. They don't eat your host tissue. They actually absorb the nutrients from your diet. So someone had this brilliant idea that, well, if we put a little packet of the eggs and somebody ate it, then one of these worms will start growing, and it's going to eat all the calories. So you could eat all the ice cream you wanted. You could eat all the fatty foods. So this is awesome. Fat banished by sanitized tapeworms. So yeah, sounds really delicious. And I'm just guessing you're not going to rush right out and get yourself a pack of these. What's kind of scary, though, is that the human tapeworms, you can get them from pork, you can get them from beef, and they can grow in you and reproduce from eating undercooked pork and beef. And next time that you go into the restaurant and you look at the bottom of the menu where it says that the government says you have to cook meat to a certain temperature, now you know why. It's to try and regulate or prevent these things from spreading. This isn't really the only place you can get a helminth. You can get it from all over the place. Another example, here's one. This is one that grows inside of a dog. So this doesn't grow as a tapeworm inside of a human. It grows inside of a dog, but you can still get similar structures. You have the little hooked mouth, you have the little suckers on the side, and they grow longer and longer. If you look at what's going on, here you have the scolex, the little head. Here you have the little sections that are full of egg sacs. Their ideal host is going to be like a wolf or a fox or some animal like that. Dogs. I don't know why I forgot dogs, but dogs. And then what happens is when the dogs or the fox or the wolf defecate, then they look for an intermediate host, which would be something like a deer out in the woods. But if it's a dog and it's around you, it becomes you. You become the intermediate host. This thing doesn't like to become a tapeworm inside of you, but it'll live inside of you in these little tiny sacs. So you have this little cyst that's packed with up to 15,000, roughly, of these little tiny parasitic structures. And they can live and live and live in you. When it goes into a deer, it grows in the deer, it becomes this, basically an egg sac, and then when the next wolf comes along and eats the deer and eats its liver or eats its brain or other parts of it where these cysts are, then it goes back into the wolf and starts the whole process over and over and over again, right? So you might be thinking, well, I'm not going to be hanging out with wolves, but you do hang out with dogs, and when dogs lick their tush, and then they give you a nice big wet kiss on the face, you just want to keep that in mind, because they can actually carry it to you, and you think, oh, geez, dog's mouth is way cleaner than a human's. I've heard that too. It's cleaner because of the acidic properties and some of the enzymes, but it doesn't mean it's sanitary or it's been completely removed of all of its germs and bacteria. So it's not sterile. Right, here's an example. So here's a picture of somebody that has one of these large cysts, and these cysts can get as big as having 15 liters of fluid around the worms. When you think of your blood supply, you've got 5 liters of blood. This is a cyst up to 15 liters. So here are some examples. Oops. Yeah. I thought I had it. There we go. Here's some examples of the little tiny cysts that have been pulled out or extracted. And here's another example of larger cysts that are being pulled out and extracted. So like I said, I'm not going to put this on a test, but I just want you to keep in mind a little bit of awareness that these things spread from one person to another. They can spread from your dog to you, and you just keep passing it down. So these tumors can grow. They can grow in your liver. They can grow in your brain. They can grow in your lungs or another common place. So here's your next research or homework assignment. You're going to search the internet for an example of each of the following. You're going to find a good example of a virus, and I just want you to write what the name of the virus is and what it causes, maybe even how you can get rid of it. Look at chlamydia. There's no big trick to that. You're going to look it up. Look at rickettsia and some of the problems that rickettsia can cause. Just pick one, or more than one. Mycoplasma, a bacteria, a fungi, a protozoa, and a helminth. So when you're looking at these different structures, or these different things, just Google it. I love Google because it takes me right to credible sources of what you want to look for. You don't want to look for, Joe Schmo that said he had a tapeworm and this is his whole experience with it. You want to look at some kind of like a .edu or a .org or a .gov are always good sources. So you're going to write these different, what are there, 8 of them here down. You're going to write down an example of it and what it causes. And here's a little quick question from material we covered last week, but an infectious organism that is most likely to alter host cell genetics leading to cancer is known as which of those things we covered before? And hopefully you know that it's a virus. And a virus can inject RNA or DNA into you. So I think this maybe is a little of a good stopping point, so you can go back, you can research those last couple pieces of material. Oops, one too far back, right through here. So it'll give you a chance to go through and look at those. And when you get a chance, after you found these answers, just come back and click on the next recording.